Hello everyone and welcome to my YouTube channel. Today in this lesson we are going to understand the insertion of protein into the ER. So first we understand the structure of this translocon. So translocon is transmembrane protein within the ER membrane. The translocon consisting of three transmembrane subunit. Okay, so it's indicated in the yellow, purple and green color. So this is the top view of the translocon inserted translocon from the cytosol, okay, showing the plug in the translocon channel. Now the second one is the lateral view of the translocon inserted into the ER membrane. So here it is the sec protein. This is a 61 protein which is present in both mammalian and yeast cells, which I have discussed in my previous video and link given in the description box. So please check and watch it. So now the insertion of protein into the ER. So integral protein are embedded in the membrane by hydrophobic sequence. They span the phospholipid bilayer. So what is the hydrophobic sequences? The hydrophobic sequence is consisting of various type of amino acid, which is the non-polar, and the mainly 9 amino acid which have hydrophobic side chain. For example, alanine, valine, glycine, leucine, isoleucine, proline, phenylalanine and tryptophan. The membrane spanning portion of these proteins are usually alpha helix and consisting of 20 to 25 hydrophobic amino acid. Now the how proteins form the alpha helix within the ER membrane. Okay, so the formation of alpha helix, they increase the hydrogen bond between the amino acid, and the hydrophobic amino acid side chain. Okay, side chain interact with the fatty acid tail of the phospholipid in the bilayer. So next is the orientation of membrane proteins. The integral membrane proteins span the membrane via alpha helix region of 20 to 25 hydrophobic amino acid, okay, which can be inserted in variety of orientation. So in this figure you can see the protein at left, okay, left and center. Each span the membrane once. Okay, this is the only one span, alpha helix once. Okay, but they differ in the whether in the carboxy C or N terminus. Okay, either the N terminal into the ER lumen or C terminal within the ER lumen. In addition, some proteins are oriented in the membrane with their carboxy terminus on the cytosolic side. Other have their amino terminus extra to the cytosol. This orientation of membrane proteins are established the growing polypeptide chain are translocated into the ER. So this is a multi-membrane spanning region in which the this is on the right side is example of protein that has multiple membrane spanning region. So now the insertion of membrane protein with the cleavable signal sequences. As discussed in my previous video, the most proteins are inserted into the ER membrane by the SRP, signal recognized particle, and SEC61 in the co-translation pathway. And this video link is also given in the description box. So this some transmembrane protein have normal amino terminal signal sequence, okay, and which is cleaved by signal peptidase during translocation of the polypeptide chain throughout the translocon. So this signal peptidase which is cleave the signal amino terminal. Okay, this is the amino terminal signal sequence which is cleaved by the signal peptidase. The growing amino terminal portion of the polypeptide is then translocated into the ER and translation proceeds. Okay, so now the translocation of the polypeptide, okay, is halted by the membrane spanning alpha helix in the middle of the protein. Such transmembrane alpha helix exit the translocon. Okay, this is exit the translocon and anchor protein in the ER membrane. The hydrophobic, okay, this is the hydrophobic. Transmembrane alpha signal is changed in the translocon, causing the membrane spanning helix of the translocon to open, allowing hydrophobic transfer domain of the protein to exit the translocon laterally. The insertion of the polypeptide into the lipid bilayer, okay, entire, and block further translocation. Okay, so this is block the translocation. And so the carboxy terminal portion, okay, this is the carboxy terminal portion of growing polypeptide chain remains in the cytosol as per the translation take place. Okay, so this is the all about today's video, and you will get notes today videos in description box. I hope this will help you and you enjoy it. Thank you.